Now let's continue on this. We're almost done. I told you this is exactly what you have to know. A lagging strand contains Okazaki fragments. A leading strand, we just have one primer where DNA polymerase 3 can just synthesize one way. But we've only talked about this direction. Well, that's good enough. As long as you know this, you are pretty much set for your exam. You should be fine. But I don't want to stop right there. I want to actually give you one last picture so you can finally see. Please don't forget this DNA pol 3 is from the 5' to 3'. That's how it synthesizes it. So now let's see. Let's look at a bidirectional double-stranded DNA. And let's try to do this. So, where's my black marker? Okay. 5' to 3'. 5' to 3'. If my origin of replication, my replication fork is right here, I'm going to start separating this way and this way. So what will I end up looking like? I will have something that looks like this. Here's my 3'. 5', 3'. And now this, we're going to pretend that this is going this way, and this side is going this way. And let's take a look at each direction and see if we can actually properly place the primers and see which side would be lagging and leading. This is a good practice example for you guys. So let's see. Let's take a look at this. Well, let's take a look at this because we've already done this. 5' to 3'. Where do I put my primer? In this region right here. Where does my primer go? 5', 3'. That means my primer has to go where? Over here. I'm going to label my primers 5', remember? So if this is the 3' region, this will be my 5' region. And DNA pol 3 is going to come in and is going to synthesize this way. And of course, as it keeps going this way, it's just going to go down, 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 down. And you have a very nice replicated complementary strand. Fair enough? Yes. How about this? How about this side though? Well, if this is already the leading strand, then obviously this is a lagging strand. We will have some multiple primers in this case. Well, let's see. 3' to 5. So 5' to 3'. Therefore, my primer must be here. My primer is over here. And DNA pol 3 is going to synthesize this way, and then as it keeps going this way, I'm going to add additional primers. So this is my lagging strand, this is my leading strand. Considering that we're going this way, helicase is going this way. Now let's take a look at it this way. Is it the same? Well, let's see. 5' to 3', 5' to 3'. Where does my primer go? It goes right here. Five prime. DNA pol 3 attaches, and it synthesizes. Complementary strand. 5' to 3'. How about here? Well, let's look at it. This is going from 5' to 3'. Therefore, my primer must be here. We're going to label it 5'. This is my 5' end, and DNA pol 3 is going to come ahead and synthesize this way. As helicase keeps unwinding this way, what's going to happen to this strand? Well, it's just going to replicate very, very nicely. This is called my leading strand now. Notice, it's different, it's switched around. Leading strand. And this, of course, is going to be my lagging strand. As it keeps going this way, now we have to place multiple primers over here. We're going to place multiple primers, assuming that this was already broken apart by helicase, and we continue in this fashion. This would be noted as the lagging strand. Now, primers contain RNA segments. Do you remember your teacher telling you that when DNA is replicated, we also have some portions of our DNA with RNA? No. That's not the case. What happens to these primers? Primers are short segments of RNA. Well, these primers obviously have to get removed. So an enzyme comes in, DNA polymerase II comes in, and it removes these RNA primers. They remove this RNA sequence and replace it with DNA. So that solves that problem. So pretend now that these are no longer RNA based. But we have these Okazaki fragments. Here we have one nice fragment, but here we have multiple fragments. What are we going to do? Right now they're kind of broken apart into a few pieces. What do? We have to ligate them. We have to ligate them together so we have one nice strand. So you need one final enzyme called DNA ligase. So DNA ligase will phosphorylate the Okazaki fragments together. Remember in my other videos, I'm going to write phosphorylate with this symbol because I'm very lazy. DNA ligase will phosphorylate the Okazaki fragments together. Okazaki fragments together. Or it will ligate, whatever you want to use. And now, what do we have? Well, this is what we have now. Let's take a look at what we actually have. So, I'm going to use different color markers. Pay careful attention. Assuming that we've completed all this, and we only looked at one direction, this is what we have. Okay, so 5' to 3'. And our bottom strand over here would be 5' to 3'. And we would make a new strand eventually. So we would make a new strand, we would make a new strand, we would make a new strand. And this would be 5' to 3'. And we would make a new strand here as well. 5' to 3'. This is how we replicate our DNA. If you look at the first example we did in the last video, this is exactly what you would get at the end. So we've taken a double-stranded DNA and we've now made it undergo replication, and now we end up with two double-stranded DNA strands. We have two. We have just now replicated this entire sequence. Let's take a look at the semi-conservative theory. Let's try to explain this. Semi-conservative theory, I believe we explained that in video 1. I told you we're going to come back to it now. Now let's take a look at it. These were the original strands that we want to replicate. So technically this is the old strand, and technically this is the old strand. Yes or no? Right? These are both the old strands, right, from the original DNA strand, and we put them under replication, and now these green ones signify the new strands. New. This is exactly what the semi-conservative theory is, or semi-conservative model. DNA replicates in a semi-conservative fashion. Old strand, new strand, old strand, new strand. Old and new. Very easy. So now we have to let's nitpick on the little things about replication. So this is basically the general picture. Brief overview. Helicase and top-wise telomerase works together. Primase lays a primer down. DNA polymerase III comes in and synthesizes from the 5' to 3' fashion. We're going to have a leading strand and a lagging strand. The lagging strand contains multiple fragments or multiple primers, and they're going to have multiple Okazaki fragments. The primers will be removed, replaced with DNA, and now we're going to have DNA ligase to phosphorylate the Okazaki fragments together. So we have a nice little thing over here. Now, in another video we're going to talk about telomeres and telomeric sequences, but let us continue. Let us continue talking about the little things that we've missed out in replication, such as accuracy and speed. How fast does DNA replication occur? I just told you we have thousands and thousands and thousands and thousands of bases to replicate. I believe that replication occurs. We are able to replicate about 2,000 nucleotides per second. This is incredible. Every second we're able to replicate about 2,000 nucleotides. That is really, really, really phenomenal. So the speed is incredible, but now comes the accuracy. How accurate is it? Obviously, if it's not as accurate, we will start to see problems such as cancer. Once again, we're going to talk about cancer in another video. But let us see, what are the rates of mutation? What are the rates of errors? And you have about, when you start to replicate, you have about 1 in 1, for every 1,000 bases, you have about 1 error. This is very bad, by the way. This is incredibly horrific. But we have mechanisms that prevent and fix all these errors that we do. And one of these mechanisms is actually the DNA polymerase III itself. And I told you that DNA polymerase III synthesizes from the 5' to 3' fashion. I told you never to think about it in the opposite direction. But now I'm going to tell you something very special. I'm going to tell you that the DNA polymerase is considered a 3' to 5' exonuclease. DNA polymerase III is a 3' to 5' exonuclease, meaning that it can remove a base and fix it. And you might say, hey, why do we want to remove it? Why do you want to go backwards? Well, perhaps you've made a mistake. Perhaps as you were synthesizing a TTGC, as you were synthesizing, accidentally you've made an A here, and you're supposed to put a cytosine here. Well, then DNA polymerase III perhaps might go backwards and say, hey, remove it and put a C here. It's considered a 3' to 5' exonuclease. It could remove a correct base sometimes, but more than often, DNA polymerase III, when it moves back, it's usually to go ahead and fix an error that is made. And this is really fascinating, and it takes the accuracy to about 1 error in 1 million bases. So you can see, just because DNA polymerase III is able to do this, we've now limited the amount of error in our replication, when replicating our genome, into about 1 in 1 million. Which is decent. You know, you might say, hey, wow, that's very good, right? This is so accurate, impressive, I'm impressed. Well, I'm not so impressed, because this is still very dangerous. 1 in 1 million bases and errors is not that great. And it's not that great because, and it's been scientifically proven that there are many people that don't have this ability of this 3' to 5' exonuclease activity, is that they most likely end up developing hereditary colon cancer. We're going to talk more about that when we talk about cancer. But if you're not able to do this, your chances of getting cancer, or specifically colon cancer, are very, 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 very high. So, what do we do? Well, DNA has proofreading mechanisms. We have proofreading mechanisms, and these mechanisms are called the mismatch repair systems. So we have the mismatch repair systems, and these guys tend to recognize. We're going to have something that goes along and starts to feel, hey, do these bases feel right? Do these bases feel right? So let's pretend I have my double-stranded DNA over here, and for some reason, because I've put a wrong base in it, there's a little bulge in this area. There's a small little bulb in this area, and this certain enzyme, this specific enzyme, is going to go ahead around and try to feel for all the bases. Hey, good job, good job. Hey, look, I feel something weird over here. Something's bulging out, something's sticking out, and obviously that's an error, so now we have to repair it. And what happens? They repair it. And now we've basically limited this into 1 in 10 to the negative 9th bases, which is basically 1 error per genome. 1 error per genome. Now that is very, very impressive. We've limited the, we've basically made this process very, very accurate. The error rate is very low. And now we can feel happy. Now this is not going to cause cancer, but this mutation rate will. So we've talked about the mismatch repair systems, we've talked about the errors, efficiency, and speed. Now let's talk about simple mutations, like insertions, deletions, missense mutations, and so on. Let's focus on mutations for a bit. As you learn when you learn about transcription, you're going to learn that every 3 bases codes for a codon, and a codon will code for an amino acid. I will reinstate this once again in the next video, but keep in mind right now, 3 bases represent a codon. Each codon represents 1 amino acid. But if we have mutations, once again, so amino acids will eventually make proteins, and these proteins, of course, are fundamental. What happens? My ribosome, so when we talk about translation, when we talk about making the actual protein, ribosomes, the organelle ribosomes, are going to read this mRNA transcript, and they're going to read codon from codon from codon. But what happens if we have mutations, right? So if we have mutations, so let's just say, let's pretend that we have, let's look at a DNA strand. It doesn't matter, we don't have to even talk about mRNA. A, T, G, G, T, A. If I have an insertion, if I have an addition over here, if I accidentally add another base and make it a C, what's going to happen? Instead of it being a codon of ATG, it's going to be now ATC. And then GGT, as opposed to ATG and GTA. Look at the differences in these. Look at the differences in these. I know this. I'm talking about the DNA strand. I don't have to talk about the mRNA strand, but you can still get the idea that we've now significantly changed this entire instruction. This is called a frameshift mutation. This is called a frameshift mutation. When you change the reading frame, right? So instead of reading ATG, GTA, TAG, you've actually changed the reading frame of it. Now it's ATC, GGT, ATA, GCC. Oh, look. It was perfect before. Now you just changed the entire frameshift, and now you're coding for something completely different. This results when you have additions or deletions of nucleotides. Similarly, instead of adding a C, what if I remove the A? Now it's TGG, TAT, AGC, CT. Oh, it's going to go on and so on and so on. But obviously, this is not good. We have a frameshift mutation. This mutation will lead to the entire sequence of instructions being changed. Now, another one is called the nonsense mutation. We've talked about adding nucleotides. We've talked about deleting nucleotides. What happens if I substitute a nucleotide now? So instead of having ATG, TAC, instead of having T, I'm going to substitute this now for C, right? What happens when we substitute it? Now, well, nonsense mutations usually lead to a stop codon. Nonsense mutations lead to a stop codon. Remember that. So we have frameshift mutations, we have nonsense mutations, and now we have a missense mutation. Which is similar? Missense. And the missense mutation, once again, you have substitution. However, it may or may not code for a stop codon. It might not even change the protein function, or it might. It might critically change the protein function. You don't know. We don't know. But this is what a missense mutation is. So we've talked about frameshift mutation. We've talked about nonsense and missense mutation. These are some of the mutations that you should be aware of. In the next video, we're going to talk about transcription.